So we are studying metabolism, and so far we have covered stage 2 of carbohydrate metabolism, that is glycolysis and pyruvate dehydrogenase complex reaction, correct? So we have acetyl coenzyme A in our hand, and which is ready to enter the final stage of metabolism, or the stage 3 of metabolism, known as Krebs cycle, on the basis of the scientist who discovered this, or simply citric acid cycle on the basis of the first intermediate of Krebs cycle.
So the overall view of this cycle, you can see from here, is that the 3 carbon pyruvate formed in the glycolysis converts into a 2 carbon acetyl coenzyme A in a decarboxylation reaction catalyzed by pyruvate dehydrogenase complex. We have covered that. The 2 carbon acetyl coenzyme A now forms a 6 carbon intermediate, then converts into a 5 carbon intermediate, then converts into a 4 carbon intermediate, and finally the 6 carbon intermediate again replenishes. So it is a form of cycle which gives CO2, ATP, and in sometimes GTP, NADHs and FADH2 on the way, correct? So that's why it is known as cycle. So the point to be noted here is that all the enzymes of Krebs cycle are found in mitochondrial matrix, because this reaction happens in the mitochondrial matrix, except succinate dehydrogenase, which is found in the inner mitochondrial membrane. Succinate dehydrogenase catalyzes step 6 of this cycle, and we will discuss this enzyme in details when we study electron transport chain, correct? So now it's a time to discuss step by step reactions of this cycle, starting from the step 1, which is known as condensation, or more commonly aldol condensation. Citrate synthase is the key enzyme of this step. Synthases, whenever we talk about synthase, you should know that synthases are ligases, because these causes the joining of 2 molecules, and their beauty is the formation of carbon-carbon covalent bond, correct? So this is aldol reaction followed by hydrolysis, and it is one of the few enzymes which causes major conformational change when substrate binds. We have discussed hexokinase, which causes conformational change. We have discussed triose phosphate isomerase, which causes major conformational change, and this is now the third enzyme which you are studying, citrate synthase, which is a key enzyme of this reaction, or of this cycle, which causes a major conformational change. So when you look at the reaction, you will get to know that the 2 carbon acetyl coenzyme A reacts with 4 carbon oxaloacetate to form a 6 carbon intermediate known as citrate. The delta G of the reaction written below is minus 32 kilojoule per mole. If you remember the delta G of ATP hydrolysis, it is nearly about minus 30.5 kilojoule per mole. What I want to say that the free energy released in this reaction is more than the free energy released in hydrolysis of ATP, and in biological system minus 25, minus 28, or more than 30 in negative terms. Whenever these number you will encounter, you will get to know that, or you must assume that these reactions are highly exergonic, because minus 32 in biological system is a big number. In negative form, it means it's exergonic. Highly exergonic often represents that it is metabolically irreversible, because we have discussed previously that great number of minus delta G is always represents a irreversible reaction. You cannot go back when you form the product, because the delta G is highly exergonic and negative. Okay. Since it is highly exergonic and irreversible, hence this is committed step, because if the product is formed, you cannot go back, because this is irreversible reaction. Why it is irreversible? Because delta G is great negative or large negative, correct? So we have understand these 3 terms with respect to this enzyme. Now the question in our mind is, from where this energy comes from? So, I want to make you aware that wherever thioester bond is present, the molecule is said to be a high energy molecule, because the cleavage of the thioester bond release a very great amount of energy, and it is because of the presence of thioester bond in acetyl coenzyme A molecule, the reaction is highly exergonic, correct? Now, it is very important to understand the reaction mechanism of citrate synthase. Do you remember this figure? Can you correlate this figure which is written on the top? Yes, in the previous lecture we have studied sequential mechanism, and somehow it represents the Kleinen notation we have just discussed. So, this is your enzyme citrate synthase, and this is your first substrate oxaloacetate, this is your second substrate acetyl coenzyme A, and third H2O binds. What is going to happen next? Release of products. So, if we see step by step, we will get to know that the binding of oxaloacetate on citrate synthase takes first. So, when oxaloacetate binds citrate synthase, it causes a major structural change in the enzyme, turning it into a closed form, from open to closed form. Changing the conformation from open to closed form happens in such a way that oxaloacetate leaves a binding site for his fellow acetyl coenzyme A, which is going to bind next, or which is just waiting outside. Okay. So, now acetyl coenzyme A also comes and binds and forms citryl coenzyme A, an intermediate. What is citryl coenzyme A? Citrate synthase in combination with acetyl coenzyme A on one hand and oxaloacetate on the other hand, correct? Now, you have citryl coenzyme A in the active site of the enzyme. Now, enzyme finally opens and let water in. So, if we do a quick recap, what happens first? Binding of oxaloacetate causes enzyme to convert into closed form, and formation of citryl coenzyme A causes enzyme to open and let water in. Why it is important to H2O come in? Because it causes the hydrolysis of the thioester bond of acetyl coenzyme A to release coenzyme A SH and citrate, the final product of this reaction. So, this is a classic example of ordered sequential reaction in which all the substrates and all the products bind and release in a sequential manner, correct? But this is not the end of the reaction mechanism of citrate synthase. We want to see or peep into the active site of the enzyme, that what is going to happen actually at the molecular level. So, this is the active site of the enzyme in which 3 amino acid molecules play a very crucial role, and these are histidine 274, histidine 320 and aspartate 375, correct? What happens first? Oxaloacetate is activated by transfer of a proton from histidine 320. So, first oxaloacetate binds the blue moiety. When it binds, or when it comes into the active site, it comes into the close proximity of histidine 320. And when both these moieties come together, what happens? Histidine 320 transfers a proton. Oxaloacetate is activated by transfer of a proton from histidine 320 to its carbonyl carbon atom, correct? Now, what happens next? Now, acetyl coenzyme A binds second, the pink moiety. This is your acetyl coenzyme A. Now, it comes in the position, and its position is near to the histidine 274, the lower histidine, correct? What happens next? The histidine 274, below, what it causes? It causes donation of a proton to this carbon, or the carbonyl carbon of acetyl coenzyme A, to form a new bond, that is COH bond, in which the H comes from the histidine residue, where histidine acts as an acid. It is donating a proton. Now, what happens next? This double bond, the carbon-carbon double bond in the acetyl coenzyme A molecule, causes attack on carbonyl carbon of oxaloacetate, which ultimately results in the formation of new CC bond. So, which actually represents the condensation reaction. Okay. Two molecules come together in the proximity of the enzyme, and due to the effect of two histidine moieties, what happens? The formation of new carbon-carbon bonds takes place, and now you have a 6 carbon citryl coenzyme A intermediate in the active site. So, it is not the final product, because the final product would be your citrate, but citrate is here along with the thioester bond or the SCOA group, correct? So, now there is requirement of water to come into the active site and do the hydrolysis reaction. So, the formation of citryl coenzyme A complex induces a further structural change in the enzyme, and enzyme becomes first completely closed to form this product completely or the intermediate completely, and then opens to let water in to catalyze the hydrolysis of thioester bond. And it is the hydrolysis reaction, when it happens, it causes release of coenzyme A SH from the molecule, and the product citrate, which is formed in the active site of the moiety, also releases in the form of citrate. So, why it is important to close the enzyme conformation when water is actually allowed to move in to further undergo the hydrolysis of thioester bond? It is because water, if allows in previously along with oxaloacetate and acetyl coenzyme A, it can cause hydrolysis of thioester bond of acetyl coenzyme A before the final intermediate citryl coenzyme A complex forms, or before the final product citrate is formed, correct? So, it is very important to allow water inside the active site when it is actually important, and you have discussed a similar kind of reaction when we were discussing hexokinase, where water is actually prohibited. But if water can enter the active site of hexokinase, the product glucose 6-phosphate in the glycolysis cannot form, and if water enters into the active site of citrate synthase complex prior to when it is required, acetyl coenzyme A got broken down, and cells cannot afford these loss, because formation of citrate is the prerequisite of the Krebs cycle, because it is the committed step, and it is the formation of citrate which actually paves the way to the further steps of citric acid cycle, and you can get ATP only when you can get citrate. So, cells cannot afford this breakage of acetyl coenzyme A molecule. That is why water is not allowed to go in previously when it is not required. So, it is a classic example of ordered sequential reaction, and it is a classic example of induced fit model of the enzyme substrate binding. So, I am ending this lecture over here, because I have discussed a very detailed mechanism of this enzyme, and I want you all to revise this very clearly and keep every step in your mind, correct? So, happy learning.